Well, I got interested in science really as a kid, and I was very excited about biology. I loved animals, I loved living things, and I was always curious about them. And then in high school, I got introduced to the idea of biochemistry and the molecules in life, not really through schoolwork, but through reading that I did. And that entranced me because I had a feeling that somehow, if you understood how the molecules in organisms worked, you would somehow understand how biology and life worked. So I went to Fred Sanger's lab and got very interested in DNA sequencing in the very early days before Fred and others, had developed DNA sequencing methods completely. And what you could sequence was the ends of DNA molecules around the time that I left as a graduate student from Fred's lab. So I thought, well, wouldn't it be interesting to find out what's at the ends of the linear DNAs of chromosomes? So back when I started my postdoctoral work and decided to work on sequencing the ends of chromosomal DNAs, there was no methodology for DNA sequencing as we know it now. And my advisor in Cambridge, where I'd done my PhD, this was Cambridge, England, Fred Sanger, had been developing methods for sequencing DNA, but they were by no means fully developed. And so when I went to Joe Gall's lab as a postdoctoral fellow, I decided to try and sequence DNAs that were from actual chromosomes in actual nuclei of actual cells, not just viruses.